Yes, we can
Yes, we
see it.
can.
Yes, we can.
All right, thank you. Good afternoon, everyone. Thank you for inviting me to this webinar, and thanks a lot to the organizers. I am going to be giving a very interesting talk to me and to a whole lot of people globally now with respect to what we are doing in breeding for climate resilience and in improving photosynthesis. Uh, and this is what I've been doing for the past 5 years now, right? 5, 6 years now. And I'm just going to be sharing the journey, how it began and what we are doing, what we have done, what we found, and what is also ongoing. And I hope I've received some questions already, and I've tried to kind of make the presentation to address some. But after the talk, I'll be happy to have questions, ideas, and also suggestions from everyone. And I also hope that I'll be able to inspire a number of people to delve into this very complex area of research. Yeah, so I'll just go straight away. Yeah, so I'm getting the feedback of people talking. Yeah, it's fine. So the reality before us globally, we know the population is increasing, and it's not that it's going to stop. It's going to keep increasing. If you look at the blue line above, you see a whole lot of trend going up, and we are still just in 2026, and it's already predicted by HONA in 2015 that global food production is going to, we will need to increase global food production to be able to meet the demand by 2050. So it's glaring, and for some of us that are in Africa here, we know a whole lot of things are going on. The population is growing. Nigeria is already eating, if not statistically, between 180 to 200 billion. And that alone, we have not even been able to solve a whole lot of issues with respect to feeding ourselves. And that comes with a whole lot of duty for us as researchers to now start exploring how to really address this food demand with respect to the increasing population. Uh, and apart from the population increase, we also know some of us, a lot of us, will be familiar with climate change, which has been, it's real. It's not just politics, even though some countries tend to politicize that. But climate change is quite real, and just this figure for the global temperature change by E.D. Hawkins, we see the change between 2000 to 2024. You see that temperature, we've had about almost 2 degrees increase in temperature. And that 2 degrees is quite something significant because it affects a whole lot of things with respect to climate patterns itself. And aside that, drought, also this is a study, this is a report from I saw from ABC News. And if you look at the heat map itself for Africa, it's going to be quite much more devastating. And this quite requires us to explore a lot of novel approach to for crop improvement so that we'll be able to meet the global food demand. We know we can only achieve, I think this is a saying from a notable person. Yeah, I think that's from, what's his name now? Yeah, Norman Borlaug. You can only achieve peace through food security. You can't keep fighting war and also having issues with food security, and you expect a whole lot of changes in the entire economy. So even as much as we try to drive peace in the global economy, also we need to also focus on crop improvement strategies as researchers, agronomists, plant breeders, and to ensure that the global food demands are met. Yeah, so let me bring it down to something more closer to us in Nigeria here, where I'm from and where I work. So you see also again in Nigeria, the same climate change also happens, especially from our head. Also, these are real data set from E.D. Hawkins from the University of Reading, and this had been a lot of predictions going on. You know, there's been a whole lot of stuff many years ago that global warming, but we didn't really pay attention to that. If you look at the figure itself, you see even in some years some period, you see some kind of peak of high temperature coming up. But apparently, everything became more glaring from 2000 upward. And I just want to narrow down to one of my field sites here in Kano, in Minjibir. You see a whole lot of increase in temperature. This is data from last year. During our field trial, it's for the first time we're recording almost about 42, 43 degrees in high temperature, with night temperature even reaching up to 30, 32 degrees. And that's quite something very huge. It's going to, if this trend goes on in a couple of years, it's going to be a very major issue with respect to food production. And also, rainfall pattern is also dropping. Normally, you would expect that some of these regions, some of the target production environments should actually have high rainfall, but you see that some is already going at a pace, and if care is not taken, is going to also be dropping down. So there's some kind of synergistic relationship between that high temperature and erratic rainfall patterns, and that really is the major reality before us, and why we are also exploring photosynthesis as a whole. So I'm going to share some crop improvement efforts from our end to actually address some of these drought and heat issues, these high temperature. I'm sure some of us would be aware. Last year, there was a whole lot of uh, information from the agency, from the meteorological agency, that there's going to be some flash floods, there's going to be high temperature, and there's going to be a whole lot of high heat. You know, even this year alone, in some of these regions where we have our few trials, we don't expect rains until around late June, July. But last year we had rains coming early as at late May, and this year also in some regions very close to us, just this week, we got information that it has started raining even in April. So what this tells us that the cropping seasons are changing, and improvement efforts are really focused on breeding for drought, heat, stress. But with this climate pattern changing, you definitely need to come up with a more novel strategy. So I'm going to be focusing on the crop that I love very dearly. As for some of us that know me, know what I'm doing. I love coffee a lot, not because just for research. I eat coffee a lot, and I'm going to share some of the crop improvement efforts, and then narrow down to what we are doing for the synthesis. This is just an overview of the target production environment for coffee, from the Guinea savanna down to the derived savanna, Sahel savanna, and the humid forest. You have a whole lot of production going on more in the Sahel and Sudan savanna, with some in the Guinea savanna. So this kind of production environment actually decide how the breeding approach goes, because some of these regions you have some of them you have rainfall pattern, and annual rainfall ranges between 300 to 400 per year, while in some you have in the Guinea savanna, you have more rainfall compared to the Sahara and Sudan savanna. So the breeding effort, the breeding approach to those regions are quite different, and that tells you the kind of variety you develop to release to farmers in different regions of production. So I just want to, I'm building something up, and I just want to share what a breeding pipeline looks like, and I'm just going to be taking a cue from Calvin. So I work with the gene bank at IITA, and you have a whole lot of novel genetic resources that are conserved in the gene bank. Basically, the gene bank is not just for storage, it's not just for conservation. You definitely want to utilize those novel sources in the gene bank, and which actually makes a whole lot of sense with respect to crop improvement. The gene bank serves as the backbone for food security, not just for conservation alone. Plant breeders can actually tap into those novel and rich diversity in those jam classes. Yeah, we do have a whole lot of crops conserved in the gene bank, but for this talk, I'll just talk about cowpea. Currently, we have almost about 70,000 accessions conserved. Now, in those jam flowers, definitely you want to identify a whole lot of things. And what we do in the pipeline, you would definitely do a lot of pre-breeding research activities where you study the diversity of those lines, then you select potential or desirable lines in the gene bank, then you move them forward. In the process of selecting and moving forward, it's what is called discovery. So basically, you not just only focus on the genotypes alone or the accessions in the gene bank alone, you want to focus on useful traits that are in the germplasm itself. Are you able to mine useful alleles in those pool of germplasm, and those traits that you found in those germplasm, can you deploy them into the breeding pipeline? Basically, that's what is called trait discovery. So in trait discovery, in the product development pipeline, you definitely have a whole lot of focus. And for cowpea, these are just some of the traits we do focus on. And so from there, once you know that the trait is very heritable and correlated with your trait of interest, perhaps yield, which is the main end goal for every breeder, then you know you can deploy that trait, including those lines, into the breeding pipeline. And for cowpea, we do have the short, medium, and late duration product development pipeline. And in those pipelines, you do have, then you move from line development down to variety, release testing, and release to farmers. So that's just a kind of overview of what happens in breeding pipeline for cowpea, which we do for cowpea improvements at IITA. So in this pipeline, you see that most of these traits I put in there, you wouldn't see photosynthesis there because it's actually it's not a trait people tend to focus on because it's complex. And even we learned from the Bambolog how evolution had really left and had really help us to discover crops, help us to discover novel traits. But out of all those traits of interest, photosynthesis had been underutilized itself. And so this is where my research comes in. Do we have very interesting traits, or do we have very interesting lines that have high photosynthetic efficiency in the germplasm that we can now put into this pipeline? Then we deploy them into other existing elite varieties for varietal release. So just because of the previous slide I shared, so that takes me to, if you're in a demand-led breeding program, you definitely want to focus on your market intelligence. You want to know what's happening from your product development to breed. And one of those critical points is you need to understand what works in every target production environment or target population of environment you're working with. I shared a figure earlier where you have the current market segment for cowpea across the Sahel, Sudan, and Guinea Savannah, which are the core production area for cowpea. So if you look at the Sahel, Sudan, and Guinea Savannah itself, and you look at the future segments, what is happening? What that means is in the near future, which has even started, every variety you need to release must definitely have a major trait of interest for each regions. So you don't want to go to Guinea Savannah and go and produce a variety that is not relevant to that kind of climate. The farmers, they would not buy from you. They will not adopt your variety, and those are some of those things in the CGI which is being tackled. That's what is more of demand-led breeding, where you are sure that your breeding program is definitely focusing on what will be adopted. And once variety are adopted, then it tells you that in that breeding program itself, then you are going to be having a whole lot of increase in genetic gain. But if varieties are developed, you spend a whole lot of years breeding and select phenotyping on the field, screening, testing, and perhaps in the course of 6 to 8 years, the variety is out and in those market segment, they are not being adopted, then it becomes a problem. You would definitely have just wasted a whole lot of time. And previously there used to be plant breeding is an art and science, but now in a demand-led breeding program, plant breeding is now both art, science, and the business of developing crops and ensuring that it gets to down to varietal release. So across in the future segments, you definitely can see that those that traits are highlighted red. The red, that's the climate resilient traits, which is drought, heat, and flood tolerance would become a major need. So we call it dominant traits in the market segment. So for every variety you need to release, they must have that particular trait before it's been adopted by farmers. Yeah, so I start delving into why we are studying photosynthesis. If drought heats are going to be a major trade of interest in any variety you need to release, then if we are looking at those drought as a whole, if you are just looking at productivity as under drought condition as a whole, then you are not looking at it in an holistic point of view. And I remember when I was studying my PhD, I came across this paper by the late Stephen Long, uh, where it talked about can improvement in photosynthesis increase crop yields. And this is a very significant paper that really changed my perception, and that really ushered me into studying photosynthesis because there's a lot being done with respect to that area of research, especially by Steve Long. Although it's late now, and I was happy that we met in a couple of a few years ago, uh, where we met in a meeting, and I was excited that at least I read a paper, and that kind of led me to focus on photosynthesis research. And it was like, oh, Sansa, you should find a time, we should plan a time where you come and let's see what you're doing on copy. And unfortunately, the time it didn't happen. But I was happy that he actually made a whole lot of impact. This is one of the leading researchers globally in photosynthesis research. And his paper has been a major focal point for anyone studying photosynthesis. And now the question is, can improvement in photosynthesis increase crop yields? And those were the research questions I was exploring during my PhD, studying photosynthesis under drought and heat conditions and trying to see what's the link between you selecting a line that is productive and what's the link between you selecting a line that is resilient. So yeah, why photosynthesis is crop prevent efforts. So in a basic photosynthesis equation, you have the light, the water, and the CO2, which gives you the crop. But apparently, breeders we could focus on, oh, we want to screen for lines that tolerate high temperature, and as most of the effort has also been breeding for water use efficiency lines that actually can perform under drought condition. And you know, there's not been a whole lot of effort around what happens with respect to the physiology itself. How do plants really transform this CO2 through Rubisco and convert it into sugar that then leads into the sink for the plant, where in context that's the grain yield for grain crops, while for tuber crops that how do they transform from that their source to sink that's producing much sugar. Most of what you get is really converting those sugar into pots or into tuber. So apparently, there's not been a whole lot of effort in this research area. Another interesting thing is, yeah, we want to select lines, we want to breed lines, focusing on above ground traits. What's happening below ground traits? What's the role of soil microbiome about in driving resilience in crops? So basically, just focusing on drought and heat alone, and without looking at what happens below ground and what happens at the physiology level, would not really give you a robust variety. You would just be breeding varieties that are productive, but you won't be able to really drive resilience. And once there's a change in climate pattern, just like I shared, you're expecting rain to fall at this period and it stops raining. What you get then is most of this variety you develop in that context of that climate pattern would always fail. And yeah, like I said, it's really been focused on productivity and yield. But now we are moving away from just productivity. We want to look at how do we combine productivity and resilience, and that's the context of we looking into photosynthesis. Yeah, if we look at yield itself is a function of light, and this equation actually tells us you have the light coming in, that's the photosynthesis, and the interception efficiency. But basically, how do plants convert this light into sugar? If we're able to tap into that, perhaps would be able to really drive a lot of resilience while still maintaining our yield, the potential yield which we want. If we're able to really understand how to maximize that light intensity, how the plants actually protect themselves when they are under perhaps high temperature stress or other stress that are affecting them. How do they convert all those response into a mechanism that makes them to be more resilient under different climate conditions? So it's a combination of you trying to track for the synthesis itself while also trying to improve yield. And what happens is if you look at photosynthesis, this would really help then boost both resilience and productivity in crops. And with that, you'll be having a robust variety that will not only perform under optimum condition, but can perform under optimum and marginal temperature or soil conditions. And with that, you are sure that your variety that you are releasing or any variety you are developing across this context would definitely be a very resilient variety. Yeah, so the question is, I know some of us in our mind will say, for some of us that might have delved into this area, it's a very complex field, it's a very complex trait to study, and one of the reasons is because it is polygenic in nature. And if a trait is polygenic in nature, it's basically very difficult to study. But we wouldn't say because it is polygenic, because yield itself is also polygenic in nature. You definitely don't have a particular chain that drives yield. So it's a combination of trade, it's a combination of genes that actually drive yield. And if we look at it from the holistic point of view, okay, fine, it is polygenic in nature. So what approach can we explore to actually tap into some of the interesting findings? How do we track some of those traits of interest or genes that are driving some of these mechanism in photosynthesis? And I just want to quickly break down the basic breeding equation where you have your phenotype, which is a summation of your genotype by environment. But by the time you start coming down, you know that if you break down your genotype, you have your additive effect and your dominant effect and the interaction. And by the time you look at all these things, they all play a key role. While the environments, while G by E also play an effect in the phenotypic expression of a trait. But if some of these traits are additive in nature, then it gives you an insight that you could actually select some of these traits, especially at the genomic level, where you use genomic assisted breeding method. If you're able to really track many genes that are giving an insight into the genetic architecture of these traits, then it's possible you might be able to get something that is very interesting. And just this figure I show, it tells you that across all these quantitative traits that I mentioned and this equation, you will see that the additive effect itself is much more important than the dominant and the epistatic effect, which is the interaction effect. So the fact that it is polygenic in nature does not mean we should run away from it as breeders. What we just need to do is to actually understand complexities and try to see how to track those complexities and then move them into breeding pipelines. Yeah, and I got this slide from Dave Kramer, is the director for the John Ingalls Institute, and this is what we were discussing. This is what we have been looking at for the synthesis. Yeah, it's very complex, but if we're able to really tackle these complexities, we may be able to really improve these crops with respect to resilience. If we're able to boost those photosynthesis, yeah, if we find traits that that are of interest, those traits are driven by genetics, not just environment itself, then we may be able to really look at possible ways to improve this crop. It's quite very challenging. As somebody working in this area of research, it's one of the one of the challenging area of research that I think anyone can delve into, because if you don't design your experiment very well, you just end up taking a lot of noise, which you would call data. So this was some conversation we were having, and this is just what I decided to share this slide so you kind of see the picture we have in the long term. If we're able to really track these variations, we found genetic signals for these variations for the synthesis trait, then we have a very solid foundation to start on for crop improvement. Yeah, so I just want to share a scenario about of productivity versus resilience. This is a trial across 3 locations in 2 years, and you see across, this is the grain yield performance. You see the yield performance that varied across those environments. Yeah, you would want to say, okay, as a breeder, I just try to go for my stability. Yeah, if we go that route, definitely you would actually be losing a whole lot of information that can give you some very strong insight. This is one of my trials during my PhD, and you'll see from here there's something interesting I want to show you. This particular environment that had the lowest yield apparently was all the entire environment was actually drought, basically drought environment. But this we decided to screen those lines under scenarios where you have combined drought and heat, and you see that the yield potential in this particular environment dropped drastically compared to other environments where we just focus on drought alone. So it tells you that if I just select lines just based on stability across those environments, I might be losing some very key information for this environment where we have combined drought and heat. And most of those genotypes might actually have been select. But apparently, it's giving us a very interesting insight to see, oh, if you're studying drought alone, then you should pay attention to other climate scenarios. And that's what we are trying to do from our head. We are trying to study under real field climate scenarios. You could have intermittent drought, you could have complete drought, terminal drought, you could have high temperature, you could have low temperature. Across those scenarios, how are those genotypes performing? What's the photosynthetic response performing? Then we can now start tracking a whole lot of things. And one of the reasons for this low yield potential is because when you have high temperature for coffee, you see a whole lot of pod abortion. So you of those pods, the flower dropping off because of the high night temperature and high day temperature. So if you want to release a variety to farmers that it's not taking in consideration of this kind of climate scenario, then even if those varieties are released, by the time it gets when they experience this kind of weather condition, that particular variety will fail. And that's one of the reasons why it's important to focus on both productivity and resilience while developing your product pipeline. So yeah, studying photosynthesis, basically, you could actually study photosynthesis at leaf level, and I shared some pictures. Yeah, it's quite expensive to study photosynthesis, but apparently, thankfully to some of the innovators that have developed some high throughput devices, though they are low cost, they are still handed, but you call them high throughput because just like the mod spec alone, the mod spec can give you some large chunks of data that you would not be able to just get measuring manually or just using a normal SPAD reading. And it's low cost, but it gives you a whole lot of give you a lot of opportunity to measure at leaf level. And also you have the LICO 64800, which is the gas exchange, quite expensive. And but this helps you to really understand what's happening both at field level. It's quite difficult to use on the field, but we've seen a whole lot of labs that are using some of those things on the field. And you have the LICO 600 itself, where you could measure. That's just a fluorometer where you could measure photosynthesis both at leaf level and also under control condition. And you have a kind of phenospecs. This measures at large scale canopy level, where all those joints where you see you have a lot of sensors, a lot of cameras, and it rolls down your field at different time and intervals. So it measures at canopy level and tries to look at the photosynthetic efficiency. A whole lot of parameters have been measured. And you also have people, I know at Lacanstan, a whole lot of people where they are looking at biochemical components, also trace losing and XX is also looking at most of photosynthesis at molecular biochemical level. And because of a whole lot of things where we have this SNP-based genotyping platform now, you could actually screen your lines, use some of these low-cost devices, the multispecs, the fluorometer, and there you could do some genomic assisted approach to study photosynthesis. Yeah, so one of those ways you could also look at genomics approach. I just decided to share something like this. Yeah, you could actually screen a diversity panel, just like what we're doing at the gene bank, where we test some of those lines. And because those lines are diverse and you are sure they are diverse before you go into a GWAS for a natural diversity panel, actually. But you're sure they are diverse, and from there you could actually track if there are trade associations. We call them marker trade associations. If you could find significant snakes that are actually giving you that could give you some insight with respect to the effect size of those particular markers, can actually tell you. I shared something earlier with respect to the additive gene effect. If you have different SNP markers that are perhaps co-located or having some kind of LD with some other markers in the same chromosome region, then a combination of those SNP markers can actually give you a more larger effect and could actually change the phenotypic expression of those plants. Also for QTL mapping, you could actually, if you have biparental populations where you have your recombinant inbred lines for your magic populations, you could actually test those lines for photosynthesis, then try to track if you could find potential TCLs and perhaps understand the function of those genes that are driving this markers. But the issue had always been you would need to validate, and most of this work actually stops at identifying the gene functions. And validation is actually key to be able to, for you to be sure that you could use that in marker assisted selection. And that's what we're trying to do from our end. We found some QTLs and that we are trying to validate under another different field scenarios that's under combined heat condition. And from there, we're trying to, if those markers are consistent, then it could help us select a whole lot of genes, select a whole lot of genotypes, even from a very wide germplasm. So that in that approach, we could use marker assisted selection, leveraging on these SNP markers. Another approach where you could use is also there's a lot of going on now with GWAS and genomic selection, genomic prediction. So from the figure I shared earlier, you see for GWAS, we focus on the significant traits. So you see those two lines here, which the red line above, you see that's the buffer threshold, which for some of the for complex traits like for synthesis and some traits that are polygenic, it's very difficult for you to find those trait associations because they practically have very low effect size. So it's always very, very, very difficult for you to use buffering threshold, but you could actually relax the threshold and use a kind of the FDR threshold where you kind of relax from your buffering. So for GWAS, just focus on those significant hits and then move ahead. But for genomic selection looks at the entire marker itself, the entire marker you're working with itself. If you look at chromosome 1, you see the significant hits where you found those two markers. You see that there are also some flanking markers that are coming on board. So those flanking markers could actually have some very linkage disequilibrium with the other markers, and they could be if they are healthy, then building those catalogs of markers themselves can actually help you, can give some very strong insight to what's happening in that particular region, what's happening in that particular loci on the genome. So that's one advantage where we are also exploring, trying to use genomic selection and also genomic prediction to be able to really see, even if we phenotype a large diverse set for photosynthesis, can we help ourselves? Can we save ourselves a lot of headache to quickly look at what's happening with the entire set of markers you deployed and see if you have some very interesting hits and also markers that you can predict that would, even if for genomic prediction, even if you don't go to phenotype other lines, if those markers are actually giving you a very high prediction accuracy, you could actually use them to predict your another entire population without going to the field. So it reduces your years of going to the field. Reduces your years of selection, and it reduces your years of hard work on, because photosynthesis phenotyping on the field is quite very tedious and harsh to work with. So that's why we're trying to employ this approach to see what we are screening on the field, what's happening with respect to GWAS and both genomic selection and prediction. So this is another approach where you could use to where you could explore for photosynthesis for crop improvement. So yeah, I'd like to quickly talk basically on this slide. Designing the photosynthesis experiment is not just something you dabble into. You really need to know what you want. You need to, your objectives have to be clear. What's your breeding objective? Are you focusing on drought? Are you focusing on heat? Are you focusing on perhaps nutrition? Are you focusing on a whole lot of things? A whole lot of field of interest or a whole lot of traits of interest you may want to explore. But your objectives then guides the kind of device you use, the kind of photosynthetic traits you are looking at. If you use the multi-spec, if you use the Lycor, you get a whole lot of traits. But now you need to, that's the headache there. You need to be sure which trait is actually going to give you a much more interesting link to what you are looking for. I, as a breeder, what I've been focusing on is I want to see traits that can actually help me to drive yield. So it kind of then shapes what I design in my breeding objective. And also your measurement approach, it's quite critical if you're running a photosynthesis experiment. I spend a lot of time more designing than even just going on to the field, because you go to the field, you measure, and measuring for photosynthesis on the field, you will always have data, even if you are taking, if you are doing anything, you will always have data, but it's different for you to have quality data and then just having just noise that you won't be able to work with at the long run. And this has been some of the challenge of just yeah, using photosynthesis trade, because you need to be sure your time of measurement, because the trade itself, photosynthesis itself is dependent on light, it's dependent on environment. So a whole lot of environment interference affect the quality of the trade, and that's why we call it's a plastic trade. So you need to design a very strong measurement protocol for you to measure something very strong on your field or perhaps in your controlled environment. And you need to take care of you need to take cognizance of the protocol you're using, depending on the experiment you're working with. The protocol you are using for your study matters. If you are working on disease, the protocol you perhaps if you go to the Photosync platform or the OpenGI platform, yeah, I'm going to talk on that very soon. So the protocol you use is quite different, and if you are looking at because some traits you just want to look at gas exchange, while some traits you want to look at your photosystem efficiency, while some traits you just want to look at SPAD, yeah, and you just want to work with that and quickly move ahead with what you're doing. So you can't just end up measuring anyhow. You need to consider the timing of your measurement. You need to consider the protocol you're also looking at, and you need to consider how you measure. You yourself, you need to, you go to the field. That's what I correct a lot on the field. You want to measure your field, your plants. You need to be sure your angle of orientation with the sunlight. You need to be sure that you are not distorting your leaf orientation itself, because some of those devices will even give you your leaf angle you get. But apparently, if you have distorted all those things, it definitely gives you very low heritability. And that has been the headache of the entire photosynthesis chain, which we have been trying to solve. Also, your experimental design really matters. If you're going to run a photosynthesis experiment, I would always recommend that you talk to a biometrician or you speak to experts in that field. The experimental design matters a lot because the way you set up your experiment actually helps you to resolve all those complexities because your experimental design determines how your analytics will always go. If you are taking care of a whole lot of if you have considered a whole lot of models that you intend to use in your experimental design, perhaps you want to delve into the mixed model approach, where you look at your blues and your bloops, then you start kind of looking at a very robust experimental design you want to use for your experiment. Some experimental design can help you to measure in blocks, while some can just help you to measure, randomly. So you just need to focus, just like what we're doing now. We're just measuring some subsets randomly, while we are measuring the entire population with respect to block measurements. So that approach actually helps you to be able to resolve the complexities when you start analyzing. The critical issue for the synthesis experiment is because the heritability is always low. And as a breeder, that's what I'm looking for. If for you to be able to move a trait into your selection pipeline or your breeding index, you definitely that trait must be heritable and must actually have a strong correlation to yield. But we found that if you're able to correct some of these measurements errors, the experimental design, if you're able to really take your measurements in a more quality way, the chances are high that you actually have something very interesting at the end of your experiment. So I'm just going to share some of what we've done from our hand and improving photosynthesis in coffee. So what I've been doing, the approach is basically to combine breeding and physiology as well as genomics to be able to really decipher. Number one, we want to see which some of these traits actually have some very interesting insight to yield. And the way breeding is now, uh, we cannot continue to shy away from physiology yet. Physiology is quite difficult for some of us that might have measured. You will see that it's very difficult to really even understand some of this photosynthesis trait. Even for me, I'm still struggling to really understand a whole lot of things with respect to some of these traits because some traits I just leave them. And most time when I get to a community of some of the photosynthesis researchers, and we just laugh because you cannot focus on all. So you need to really know which of these physiology traits are quite interested, and then you can then focus on perhaps leveraging your genomics approach to drive your breeding objective. So what we have been doing from my end, so what we did, which was quite also interesting, because it's been difficult to phenotype under very few conditions. Most of the photosynthesis work actually starts from controlled environments. Yeah, there's a whole lot of efforts on that in the global space where they study photosynthesis under controlled environments, and but they found out by the time you take those things out to the field, it's a different ballgame. So what I did was from my hand, okay, if we have some very low high throughput device that are low cost and easy to handle, if we are able to really phenotype under real field conditions, then we may be able to then rank and select lines, and which we then do a more intrinsic phenotyping on that controlled environment. And that's the approach we've taken from our head. So these are some of the key research questions that it's as a breeder and you could look at for your crops. You need to know which of those traits out of the entire physiology trait, out of the entire photosynthesis trait, can actually help you to drive yield, can actually help you to select for climate resilience. That's what we are doing also. If we are able to know that stage, then we might be able to just focus on that stage of development and then put all our efforts in exploring the variation within that stage, and then it saves us a whole lot of resources and time. And you exploring those variations is actually the basis for anything you want to do for improving photosynthesis. And also because there's a lot of going on modeling, if you could actually predict canopy photosynthesis, perhaps using the LiDAR or the fellow MA vision I shared, if it could help you predict your yield. So we have a lot of expertise going on this in some labs that I'm aware of, trying to predict that to yield. And also from the genomics angle, if we're able to now see those genes, can we able to dissect the architecture of those genes? What's their mechanism? How do they differ with respect to genotypes? Are they easily introduced to other to other allelic varieties? And because as the plant breeder, you also want to be sure how you want to understand how your G by E affects your stability of photosynthetic traits across different conditions, across different environments, across different times of measurements. Once you're able to resolve that, you're able to find some lines that are promising, that have some of these interesting traits of interest. Then you could develop perhaps a biparental populations on that and start tracking down for your trait of interest. So the overall goal for me from our hand also is basically we want to ensure that we could have a selection index that actually give room for photosynthesis traits. So what I just did was, for those who are familiar, in your selection index pipeline, you actually use this made EZU index where you apply weights to your traits of interest. I won't talk too much on this so that I don't just bore people. So basically, you apply weights based on the economic importance, based on the priority of the trait, and it could be your major trait of interest in that your product development pipeline. So I just give a kind of example here. Now I have my trait of interest, which is yield, and I gave it a weight of 3. And perhaps SPAD, because it gives me an information on chlorophyll, I could use it to select large amplasm even at seedling stage. I could give it 1. And you have Phi2, which is the quantum yield of photosynthetic efficiency. I could also give that another weight. So what happens in this kind of breeding index we are looking at designing is the overall gain which you have for when you plot this your index per year. If you have very high index accuracy, then it gives you an idea that you will definitely have a very strong, a very improved genetic gain at the end of your breeding program. So this is just something we are trying to explore for photosynthesis. It's novel. It's something that has not been done, and we are hoping that we'll be able to find some traits from our study that are consistent. And after a series of measurements, we are sure that this trait of interest can actually support selection and can actually contribute to the overall yield itself. So this is just one of the work we've done, which has been published, where we screened some of those lines during my PhD under climate field scenario. So the interesting thing, which has been very important and has been driving a whole lot of global interest now, is because we're able to find some interesting correlation between photosynthesis and grain yield from our study. And we also found some markers in which, when we looked at the functional annotations of those markers actually have been functions related to photoprotection under stress. So, which is something very interesting for us to explore. So you could read, if you're interested, you could read this paper further to actually see some of the results we found. And interestingly, why this is quite much more significant for us, I mentioned the other time, which I would say we did right from our hand, was the kind of experimental approach we used for this study gave us some of these interesting findings you saw here, because we had a whole lot of mixed model approach. We had a whole lot of even designing the data collection itself was quite strong enough. We don't just go to the field. I don't just go to the field and start to measure. Some of my team, we start tracking when we have a good weather pattern, when we have a good sunlight. And if you are going to measure, it's better for you to measure some sets in a day if you cannot complete the entire experimental plot. It's quite difficult to go by with, but that was an approach we thought we could do, and that gave us some interesting results. Also, so ongoing, I shared a figure earlier where I was talking about where we had a particular environment for drought and heat where we had the low yield. So what we're doing now is we're trying to generate a golden data set for photosynthesis in cowpea. So last year, the image by your left where you have 3 guys phenotyping. So we're trying to increase the speed of measurement measuring at the same time, measuring the same rep at the same time. And the goal is basically if we are able to increase the quality of how we measure and the speed of how we measure, we may be able to reduce the environmental noise, we will be able to actually have improved heritability. And the interesting thing also is the same set that we sprayed, the same set of lines we sprayed during our PhDs, those lines are now thrown back to the field under climates, under combined drought and heat. I said the yield response we had was quite different. So we want to see if we find consistent results based on what we did earlier, which we published. If we find the same consistent result or markers that are collocated in those regions, which of what we found before, then that gives us a very strong insight to start moving into validation phase. Then we can then move to the deployment phase. So this year, currently, we've been in Kano. So it's been a very active work for the past 4 weeks. We've been measuring also. So this year, we tried measuring every week. We are measuring across the entire crop growth phase. And what we're trying to do basically is to actually track which phase which phases photosynthesis are limiting. So at this phase, we're also imposing drought. We also impose. So the entire environment is already its environment and where we have low fertility soil. So we have a lot of multi specs going on. You see a lot of people on the field where we're measuring different data points, and we're capturing as much as data points we could capture. So this is going to give us a more robust data set that could also support the genomic selection and genomic prediction we are working with. Special thanks to to these guys that are here. It's been a lot of work for us phenotyping all these things. Every day, Monday, Tuesday, Wednesday, Thursday, and Friday at times, we're always on the field between 9 to 1, 2, 3, 4 p.m. to be able to finish up because that's just the only way we could go about it. It's quite a lot of work. And if you don't have good energy, you don't delve into this kind of thing. So that's the overall goal is we want to, if we have those robust data sets under these real field conditions, then we'll be able to really track a whole lot of things for breeding. So I want to share some of the results from what we were working on, which just to kind of buttress what I was showing the other time. So from those environments, by the time we looked at their stability and we calculated their stress tolerance index, so you have some lines that are performing based on their top 20. But apparently, when we moved to combine drought and heat conditions, that was our last year trial. Some of the findings were quite different. The same line that was performing under drought alone, which is one of the highest yielding line, didn't perform under combined drought and heat condition. So this gives us a scenario that if we are selected for productivity and said, this line is promising. And interestingly, that line was quite promising across almost all locations, except for when we pushed it to drought and heat condition. So it gives us a very, very strong consideration. And also to refresh what I was saying, if you had selected this line, selected some of those baselines and you move ahead with them, under combined drought and heat condition, it's a different scenario. So what we do in the stage gate I shared, we need to rank. So we want to not just select lines that are productive, we want to select lines that are that combine both productivity and resilience. So what we did was to now look at possibly we have overlapping lines. And interestingly, we found some lines that are overlapping across those two conditions. So it is these lines then we are sure that if we validate them again in this current field year trial, then we move them under controlled condition where we could actually now study what's happening with the photosynthetic efficiency of this line under drought and what's happening to these lines under drought and heat. And that gives us a whole lot of insight where we can now start developing bioparental populations on. Yeah, so also we found some very interesting QTLs also from just our last year study. It's just a 1 year trial that combined drought and heat. We saw some very significant heats and some flanking peaks, especially for hello, which is forces excess heat and excess light intensity. So you see, if you look at chromosome 3 for this line during stage, you see there's something really happening because you have a very flanking marker pitching up. So for this trait, this is also, we found some lines that was also collocated from what we published before. So it gives us some very strong confidence that we will still be able to explore some of these markers on a bigger scale with respect to perhaps genomic selection, or then we select some of the markers that have high effect size and very strong for us to move ahead with for validation. So this is also for, if you look at SPAD under before stress and during stress, you also see some market rate associations where we found just from a single year trials using the blues. So this is some of the work that is ongoing, and we're still generating robust data sets to be able to, for us to be sure that what we're working with, because it's quite complex, but we need to validate as much as possible that what we're doing is something we can move ahead with. So we are currently on the field this year to test that. Also, what we are doing from our end, which I know some of us might have seen this, we had last year, we are building a community of expertise, but for both agronomists, pathologists, plant breeders, computational biologists, ML, and machine learning experts, yeah, because I just shared that we're taking a lot of measurements across week. That's going to be a large data set for us to work with. So as a breeder, I wouldn't want to sit down on that and start cracking my head with over almost 20 traits in a particular from a particular device. So and we have many devices which we are using, not just even the multi-spec alone. So that's what we are doing, and we at this occasion we're able to really make some very interesting findings, some very interesting approach, which will be leading to much more for us to build a very strong community, both in Africa, and it's going to be a nexus where for those of us that are in Africa, we're going to be connected with a whole lot of experts, and because we have the climate field scenarios, and we have a whole lot of things to study with respect to climate change, then we'll be able to really leverage on this community of expertise, and then we move ahead with what we are looking for. And the goal is to actually track photosynthesis to genetic architecture, because once we have that, then that gives a pathway to both gene editing approaches, both developing bioparental populations, and the selecting lines that can move for varietal release. So also, I'd like to share an open platform also from with one of our partners that we're working with. For some of us that may be interested, we may want to look it up online. It's called OpenGII. So we're trying to create a much more robust science community where you could actually measure your photosynthesis rates. This is a free platform. The Photosync platform is going commercial in a way, but you could still use it if you can afford. But what we want to do with this community is we have a whole lot of data set that is coming from different reading programs, different research groups. Then we'll be able to now bring a whole lot of experts, both the data scientists, both the machine learning experts, and then start looking at a whole lot of approach, both genomic prediction, both machine learning approach, feature engineering approach for some of those traits, and also looking at the mechanistics approach for resolving some of this for the synthetic trait, not just looking at variation alone. So this is OpenGII I'd like to share with us. So the question which some of us might want to ask, has there been any successful improvement to boost our photosynthesis? Yes, and yes, and yes. So this it's a project, if you look at it, you could read it off. PhotoBoost is a project that focused on rice and potato in Europe, which is led by Stefan. And this is a project that had increased our photosynthesis trait by yield over the baseline of 20%. And also we have the RIPE project, which was Stephen Long, the late Stephen Long started. They've really done a lot on soybean and a whole lot of looking at biochemical, studying photosynthesis both at leaf and mostly at mechanistic biochemical approach. So we have some interesting new project also on photosynthesis, which is scope that we are part of and leading for IITA with partners in Ghana across board. We also have Fabulous that's focusing on cassava, which is led by Dr. Rabi in cassava, and you also have the Suyima, which is also focused on maize and rice, led by a team in China. So this ongoing effort, because there's been existing effort that has shown that you could actually drive crop yield by over 20% by boosting photosynthetic efficiency. So, and we hope this ongoing effort also would give us those kind of interesting gain for cowpea, cassava, maize, and rice. And some of these projects are funded by Gates and Gates Foundation. Yeah, in conclusion, photosynthesis is critical. In context, even evolution has not explored photosynthesis as a target for crop improvement, and that's why it's been underutilized. And we know one of the reasons it's been underutilized, especially in the breeding community, is because it's complex. We've had a lot of headaches in the past 4 weeks of phenotyping on the field, a whole lot of issues on the field. So that's why I said you need to really be in touch with somebody who understands the field if you're delving into photosynthesis research itself. So, but apparently we are exploring a whole lot of things, and we found variations from our previous studies, and we found overlapping lines that we can exploit for understanding mechanistics approach for photosynthesis, and that could help us to really drive breeding for resilience. And I would like to really say research is going more multidisciplinary. So until we are able to really combine physiology, genomics, breeding across board, then at that point we will know that we'll be able to really accelerate genetic gains in breeding programs. And that's what we are looking at and targeting for photosynthesis. Some of the projects I just shared recently actually having those kind of experts where we have the physiologists, where we have the agronomists and the breeders, then coming back to the gene editing experts, you have a whole lot of genomics experts also looking at those approaches to look at photosynthesis, both at below ground and at above ground. And the overall goal is to actually improve genetic gain in breeding programs and also develop resilient crops for sustainable food production. I'd like to thank my team. It's been a lot of work on ground. These are the boots on the ground. We've been phenotyping under 39, 40 degrees, 41 degrees for the past 4 weeks, which is quite very difficult to do. But these guys have been resilient and they've really done a lot of work. It's not a work you could do alone. And I'd like to thank everyone for their support and also to acknowledge some of the funding that is driving some of the projects I'm running on photosynthesis, and which is funded by Cambridge Gates and the Carnegie House Institute. Yeah, thank you for listening. That takes me to the end of my slide. Yeah.
Thank you so much, Dr. Sansa. Thank you. I can see the claps going and the emojis, and thank you. It's very exciting research, and we're so happy to have you here. It's a very insightful session, and we already have some questions, and I would ask Samuel and Jimmy to unmute themselves so they can ask their questions. Samuel, are you able to ask your question live?
Yeah, thank you so much, Olakunle, and thank you so much, Dr. Sansa. I hope I pronounced the name well. I wanted to ask, I know in one of when you were trying to explain some of the breeding goals of for the cowpea, one, if I'm right, like dual purpose where you breed for both pod yield and fodder yield, like its leaves and the pod. I wanted to ask, my assumption is that there are different pathways for pod and different pathway. That's my assumption. Have you experienced a kind of like an antagonistic relationship where you're trying to breed for pod? I mean, where you want to combine both traits in one crop, in one variety, but one suffer at the expense of the other. Have there been experience, and how did you resolve that? I don't know if you get my question.
Thanks a lot. That's quite a very, yeah, I get your question very well. That's quite a very, there's always a trade-off actually. So what's, I'll give you an example. You want to develop perhaps for a target market that is looking at biomass and a target market that is looking at grain. So if your breeding objective, if you're selecting for lines, then you need to be very careful because there's always a trade-off between the two itself. So there has been examples like that, and I know if you are looking at those synergistic relationship, it depends. It's just more dependent on your breeding goal. So what we try to do to actually have a dual purpose is now you kind of run your pipeline first to identify those different lines that have those characteristics. Then you pull them together to develop. So you start crossing them together to practically have something that have that dual purpose in nature. That's why we used to say in breeding, you cannot have that super variety. So that's why breeding is always continuous, you know. So, but at times we've seen efforts from some of our breeding programs here where we have some dual purpose cowpea that actually produce much more biomass and also high grain yield production. So, but what happens is a whole lot of effort has gone into the breeding pipeline in itself during product development. You would have actually selected a line that is perhaps having a characteristics for that and different characteristics for that. Then you then start crossing them once you have you do a lot of screening. Then you have a line that actually have combines those trade of interest. So it's always a trade off. Even on the field, also you could see some lines that they just shut down and start producing biomass a whole lot. Yeah, while you have some that produce pod prolifically. So it's always a trade-off, but it's something breeders have always played around with just by selecting lines and then crossing them back together to develop something robust. I hope I answered your question.
I'll take silence as approval. So, Jimmy, do you want to go next?
Yes, can you hear me?
Yes, we can.
Okay, yeah. Thank you so much for that presentation. It was very insightful. So my question is, um, I've been doing also a lot of research work with photosynthesis, and especially for climate resilience. And one thing I realized was some of the breeding lines that I was using, even though I was using my crop was peanut, but this can apply to other crops also. So some of the breeding lines that we were using, we realized that they can be when we expose them to heat stress, they look that like they are really affected by heat stress. However, after the end of heat stress and we give them time to recover, they kind of bounce back very well. And my question is, have you ever considered recovery as part of the breeding goals rather than just, being faced with stress and leaving that alone? Because at times maybe recovery can work well. You also want crops that can recover, bounce back from stress, and be able to pick up and do well. So that's just that's what I wanted to know if you kind of have ever considered that in your experiments.
Yeah, although I kind of missed some of your point, but if I hear you clearly, you're talking about lines that you screen under heat stress, and they fail under heat stress, but once you put them on normal condition, they recover rapidly, right?
Right.
Okay, yeah. So what from in fact that's what we are doing from our current work. But what happens is you would want to you as a breeder you want to be harsh in your selection. You also want to be harsh in your breeding approach. So if a line is not able to have terminal heat stress, or perhaps cannot tolerate long heat stress, and at the same time you have a panel that can actually produce high pods prolifically under heat stress, I would go for the lines, that kind of line, because I know that even if you push that line anywhere, it will produce pods you get under heat stress conditions. So that's what we are doing, because from our previous study, we saw that some lines, so like the super line, the best uh, performing line I shared in the figure I shared earlier, you see that by the time we threw it under combined drought and heat, it didn't perform well because the flowers are budding. But I wouldn't just want to go ahead with that kind of line, even if I if I put it under normal condition and it goes back, it recovers back. You want to, at the foundation of your breeding program or at the foundation of your breeding objective, you want to select lines that are heat tolerant. You get that can produce spots that can withstand heat tolerant, that can withstand heat. And then if you have lines like that, then you can then move them ahead while you also keep track of those lines that even when they were under stress, by the time they recover, by the time you put them up, perhaps when you take up that stress, you put them back, they recover. So what happens is while recovery, and I need to mention that while recovery is quite important, and that those are some of those things we are going to be looking more at a detailed phenotyping under controlled condition. What we are just doing now is more harsh because we want to start with the best lines first. We want to start with a more stringent line before we now start looking at the approach of recovery. Because the idea is you want to select a line that can recover also, that has mechanism to recover if everything looks normal. Because under real field, real farmers condition, majority of them can plant, they can throw their lines to the field because they said this is growing season. And perhaps high temperature sets in or drought occurs. And that can just be for 3 weeks alone. So if you have lines that can't withstand that kind of condition, and they just go up. But if you have lines that have recovery mechanism, so even within that 3 weeks, they could be up. Some of them could shut down. I can see there's a whole lot of mechanism when our plant respond to some of these stresses. Like you could have the escape. Some of the lines will just close their stomata. Reduce their transpiration efficiency drastically, just to kind of keep intact, or to kind of how would I put it now? To maintain the rate at which they lose water. They just want to conserve water. So what happens is under such conditions, if you have lines that have good recovery and they are part of the lines on the field, once the rains are back or once the weather is fine, they will perform well. So we kind of select those lines, and that's one thing. You don't just throw those. You don't just go with the best lines. Also, you also have to move along with, and that's why we call them contrasting lines. You could have lines that recover. You could have lines that will fully perform, and that gives you a much more robust panel. Okay, so we look at that, and that's what we are currently looking at for our copy experiment in Kano here for drought and heat. So once we have lines that can perform under this high temperature, then we take them in control condition. Then now start designing an experiment where you expose to different time of light, different intensity of light, and recovery. Yeah.
Thank you so much, Dr. Sansa. Please, if you have more questions, I dropped Dr. Sansa's LinkedIn in the chat box. Please reach out to him. I'm sorry, we can't take any more questions. This session is recorded. I know somebody had requested if Dr. Sansa is able to share a slide, but this session is recorded and the recording will be sent to everybody when it's uploaded to YouTube. So please, I'll hand it over to Moise now. We have a few things we'd like for you to do before we dismiss.
Okay, thank you, Olakunle. Thank you, Dr. Sansa, for the insightful presentation. I think we have learned a lot today about photosynthesis. Before making the announcement, I will request if we can all turn on our camera so that we take some nice pictures. So, can we do that? Yes. Yes. I think it's okay. So let me share my screen. Are you able to see my screen?
Yes, we can.
Okay, so thank you, Dr. Sansa, as I said before, and thank you also to all the participants who joined this webinar. Were more than 100 participants, which is really good. So before ending the webinar, I will kindly ask if we can all submit our feedback using this QR code. So this one will help us improve our next webinar so that we have a very good webinars as today. So kindly scan the QR code and share with us your feedbacks about this webinar. We also have some social media platforms, LinkedIn, Instagram, and YouTube, where you can find our announcements. So kindly follow us on those platforms also. I will let this so that you can all scan the QR code and share your feedback. Otherwise, thank you very much to all of the participants. Thank you to Dr. Sansa, and I wish all of us a very good end of the day.
Yeah, thank you very much.
Yeah.